probably less clear title was my own quest for the meaningful visualization of gene expression data. So using gene ontology, as we just heard. And so being a bioinformatician in a lab of biologists, we're studying bovine immunology, the response to tuberculosis infection, a model like any other. And being the bioinformatician, I was asked to analyze the transcriptional response of macrophages to infection. And once you have the whole genome to survey, you'd like to know a bit faster which genes to look at first, what are the differences between potentially multiple conditions. And surprisingly, it was a pain to find tools to visualize gene expression profiles over time, splitting by infection. You want to follow each infection over time, and I couldn't find that. So this was essentially a toolkit for visualization of data, and then the statistics came in. So once you work with immunologies, essentially that's a typical gene expression experimental design, where essentially you take a few animal replicate cells from different animals, you expose them to different treatments, so virulent M. avium and bovis to virulent mycobacteria, BCG, the vaccine strain that most of you may know, and uninfected control. And then at each time point, you for each of those cells, you take gene expression data, and you essentially have 26,000 genes to survey at each of these time points in each animal. So how to visualize this in a proper and neat way to find biological meaningful results. So essentially, once you do, this is a micro experiment, and once you have all your probe sets here, which give you an expression value represented by color in each of your 72 samples, you have a heat map like that, which is fairly uninformative once your supervisor asks you what's going on. And essentially, when you're looking for an effect of treatment, so at the bottom line, you have this nice little flag where no one clusters. On the other hand, you have your animals which very neatly cluster, and you ask, okay, what's going on? I'm looking for differences between treatment, and this is telling me nothing about it. So why would there be a clustering by animal instead of treatment when you expect the biological effect to be more? So, or why is this heat map so blurry? You have multiple experimental factors, so you've got interactions. Maybe your BCG infection at 6 hours looks like your bovis infection at 2 hours, and you need to normalize that. There's some good time series software for this, but in this case, it is just very annoying with this heat map. You've got noise in data, you can't avoid that, but since it clusters by animal, it means that some signal, some biological signal goes beyond the noise and is meaningful. And then what's actually interesting is that the clustering, once you have a clustering, it's one result, which essentially can be driven by a few genes of large effects, while most of your genes, which may be of interest, are hidden in that clustering. So how do you actually increase the signal that you want to see over the noise or the signals that you're not interested in? So what I found in this particular data set is that I did an ANOVA to find the genes that has most variance between animals and less variance within animals. And here you can see that when you look by treatment, you have this huge variation. But if you cluster by animal, you see that animal 1 always has a low expression, animal 2 high expression, which in this case, essentially, these are immune genes, MHC, for those of you who knows, which are among the most variable genes, which makes us largely different from each other. So obviously, if you're looking for an effect of treatment, and you have those genes in your data set, your clustering will be driven by the, sorry, what I should say is that probe sets on microarray have a specific sequence. So if you have the reference genome or close enough, you will be measured and have high expression of it. If you don't have the reference genome, even though you express the gene, it might not be picked up. So that might be artifacts that you're not interested in. So how do you get rid of that noise and focus on what's really meaningful? And if it's only one gene, it would maybe be okay, but if you have dozens of genes, all of those are MHC, so have the same different patterns, but essentially differences between animals, and you just say, I need to get rid of those to focus on what makes the infection behave differently. So the question leading to the package I'm going to present and hopefully demonstrate is, can we prioritize gene classifying the phenotypes of interest, and potentially multiple? If you have only 2, you do differential expression, and that works. But if you have multiple ones, you need a sort of classifier approach. And once you've ordered, you do that on a gene level. And once you have your list of genes, you might actually want a panel of genes that is robust, that tells you something about the biological story of it. Because you don't want to go through 26,000 genes with a pile of paper showing to the supervisor, let's go through gene 1, 2, 3. So can we find a biological function or cellular processes that are robust? And I'll come back to that. So essentially, GOexpress right now, to classify genes, to find the genes that classify a treatment, for example, or your phenotype of interest best. I told you about analysis of variance. Historically, that's the first one I've tried just as a ad hoc solution. But our current favorite is the random forest. So essentially, this builds a classification tree. So you have all your genes with the expression value. And what a random forest does is it builds many classification tree, but each of them, at each node, it will tell, okay, which of the genes in a randomly sample panel of genes classifies your 4 groups, BCG, your 4 infections best. And then at the next node, you say, can I find another gene that further purifies the node so that classifies them even better based on the classification already reached? And it does that until it can't find a gene that actually makes the classification even better. And it does that many, many times, randomly sampling genes. And then you have a classification power or a score for each gene, which tells you how best is the clustering after using this gene than before on average. And the ANOVA, most of you might know, it essentially maximizes the variation between group as opposed to variation relative to the variation between group. And this way, if you have 4, if group 1, you will find the genes that maximizes those distances. So the one I'm going to present, I'm going to use in the demonstration is the random forest. And once you have your genes that are classified with the classification power, you can actually rank them, which is a bit more stable than classification power, because random forest, it's a sort of bootstrap approach, which doesn't give you a p-value, doesn't give you, it really just tells you if a gene classifies better than another. So the best thing that I was advised is to rank your genes. And then if you want to find some common biological theme, one of the approaches, which is a bit raw but works, is you score each of your gene ontology terms using the average rank of the genes. So essentially here we're working with immunity, and it's no surprise that the chemokine activity ranks better on average than the kinase binding, which is still good. But in this case, yeah, the best one is chemokine, and I'll come back to that. Also, a drawback of this score is that Go terms can have some of them have 1 gene, some of them have 6,000 genes. So when you take the average, an average of 5 is 2. The best average rank for 5 genes might be 2, and for 6,000, if you take the average of 1 to 6,000, you get 3,000. So you're not scoring on the same scale. Okay, 5 minutes. And so this is a limitation, but I've implemented a filtering function which in this case, I filter for at least 15 genes. So you've got all the 15 gene Go terms competing against each other on the same level. And this allows you as well, if you filter for increasing numbers of genes associated with an ontology, 5 gene will be a very specific, like regulation of apoptosis, while 6,000 usually will be genes annotated to cytoplasm, which is not the most helpful. So you want to go with a finer granularity to have something specific and increase as much as you need until you just get something that's not very informative. Anyway, so back to the experimental design that I'm going to demonstrate GOexpress on. You've got 4 infections, 5 animals after filtering, and 3 time points post-infection and 1 before infection. What you typically have in transcriptomics datasets is normalized expression data, filtered for genes that are expressed above a certain cutoff. In this case, it's microarray, and then you have your phenotypic data. And in this time, you have replicated treatment time. Will be interesting what makes treatments different. So essentially, those 2 matrices. Bioconductor has a nice expression set. Now they have a summarized experiment that I'd like to integrate. But so far, it's an expression set you have to provide. Then you can either automatically download the annotations for your dataset, or I highly recommend to download them yourself from Biomart in a very simple tables, because then you can download from the version of Biomart you want, ensemble release X, and you know which annotation you work with. If you don't specify that, it will always download the latest. So you give annotation for gene, a short description of your gene, gene name, and associated with feature, Go term, description, and the Go term ID. And this will be used to annotate your results. Essentially, analysis runs in a command. You provide your data set. You say which factor you're interested in maximizing distances, your annotations. Here, I decided to work only with time points post-infection. So I filter out the time 0. You can put whatever you want here. The manual is as explicit as I could make it. And then the random forest is the default anyway. And then once the first place I go to usually is the top genes, which are the best classifiers. In this case, it's 2 good chemokines, and it goes down quite interestingly, I've shortened for the interest of time. These are the visualization function that I've implemented. Perfect. Which is, as I said, I was most interested in the visualization. So I wanted those plots, and then I implemented the statistics to find them the most robust one. And as you can see, the top genes are the ones where you 4 groups are best separated from each other. And this was using the random forest. ANOVA will give you very similar results, just with different logic behind it. So this is the top gene, and if you go to the second SAA3, the second one is actually not annotated. So, but the third one also has. So sometimes you have 2 groups overlapping. It's just as good as the data is. If your 4 groups are no different, if 2 groups are not different, you won't be able to classify them. But this is statistics, but they don't impact the expression. This is purely expression data driven. No matter which statistic I plug in up there, the plots will be exactly the same because it's the expression data that you provide. It's nothing that I compute. I don't change anything. So it's really just, this table helps you find the plots that are most interesting in your data. And once you've done that, you summarize by gene ontology. So I filter for at least 15 genes, and surprisingly, as I said, it's biased towards smaller genes, but the top one at 35 genes, which is indicative in this case, if you have more genes, these Go terms struggled more than the others to get at the top of the ranking, so it's particularly meaningful. The next one is 19, which could have been higher by chance. But if you look at chemical activity, you will see there's a block of green up there, which are the control, which cluster very well away from the others. And at the bottom, you've got the embovish, which is the most virulent infection, cluster apart. And then you've got 2 intermediate phenotype in the middle. And then also, that's also one of the plots from the package. And you can easily, in one command line, you generate those plots. And in the another command line, just one, you can say, okay, I want to only have the 2-hour sample. I only want these 3 infection groups. So it's fairly simple, and everything is in the manual. So we were talking about the command line in the keynote speaker before, and I know biologists don't always like the command line, and particularly my supervisor sends me email, okay, can I have this plot for this gene, and email after email, you just realize, okay, it's probably just easier if I use that R package I just found, where you can actually implement an interface, and you say, okay, supervisor, please connect to the web link on this web server, type your gene in the box, select the animals you want to show, select the infection groups you want to show, and you just implement as he requires every little tweak that you need. So, and this is highly dynamic. I don't have time for a video or anything. So every time you just tweak something here, the plot updates. So it's called a Shiny package in R for those who don't know. And it's really a nice dynamic interface. I will skip, these are experimental features. First time we submitted, the reviewer wanted, oh, there's no p-value in there. I want a p-value. So I implemented a p-value, but random forests are not meant for that. I can discuss that at the end, but it's a permutation based. So just to say how significant, how much by chance can this Go term reach this particular rank? And one of the interesting one also is Go terms can overlap sometimes, or you have one including the other. So this is just, all those are one-liner plots, and it tells you how many genes overlap in the top 5 Go terms, and in this case, none of them overlap. Essentially, so this is GOexpress. So I started with the functions, the plots. Then I came back to the statistics saying, okay, which are the plots most interesting to see? You can do on gene basis, gene ontology. Each of the tables can be exported, and then you can have your nice plots and write a shiny interface, pardon the pun, around it. And yeah, I'd like to thank my supervisor. All the people really helped, and a few meetings I've presented this tool to, and some of the features I've presented come from direct feedback during the talks. So I'll be happy to take question and suggestion. Thank you.